Alright, so Chapter 8 concerns material involving bacterial genetics. Alright, so the first section of material covered in this chapter has to do with DNA structure, has to do with DNA replication, has to do with transcription, and translation. And I'm going to focus on each of those topics, and you're going to have little mini lectures over each topic. Alright, so first off, I'm going to discuss DNA structure. Alright, so if you recall that when you're talking about nucleic acids, alright, nucleic acids are made up of monomers referred to as nucleotides. Okay. And these nucleotides are composed of three basic components. Alright. You have a phosphate group. Alright. You have a pentose sugar. Alright, so in terms of a pentose sugar involving DNA nucleotides, you have deoxyribose. And RNA nucleotides, for RNA, you have the sugar ribose. Now, the bases, you have pyrimidines and purines. Alright, so the bases that you find in DNA are going to be cytosine, guanine, thymine, and adenine. Now, when you're talking about RNA, alright, you have cytosine, you have guanine. Alright, so you have your pyrimidine and purine. They also have adenine and uracil. Alright, so there is no thymine in RNA. It's only going to be found in DNA. Alright, so these nucleotides. These nucleotides have a polarity. Alright, so the backbone or the rung of a molecule or a polymer, polynucleotide of either DNA or RNA is made up of the sugar and phosphate. Alright, so you have a phosphate, you have a sugar. Alright, so here looking at the primary structure of DNA. Phosphate, sugar, phosphate, sugar, phosphate, sugar. Alright, now that makes up the backbone of DNA. Alright, it would be phosphate, sugar, in terms of ribose, phosphate, sugar, ribose, phosphate, sugar, ribose, if it was a primary structure of RNA. Alright, and then your bases, your nitrogenous bases, stick out to the side. Alright, so they make up basically the rungs of the ladder. Alright, now your nucleotides and your nucleic acids have a polarity to them. Alright, so one end of the molecule, alright, you have a free 5' phosphate. Now, this phosphate has a negative charge. Alright. And at the very end of your nucleic acid, your polynucleotide, you have a free 3' hydroxyl. Alright, so the very beginning of your polynucleotide, you have a free 5' phosphate. Alright, the reason you get the 5' is because this phosphate is attached to the 5th carbon of the sugar, alright, of deoxyribose. Alright, because it goes 1, 2, 3, 4, 5. Alright, so in terms of your DNA and RNA nucleotides, they are always going to be assembled 5' to 3'. They're always assembled in this direction. Alright, so that means that nucleotides have to be attached to a free pre-existing (3' hydroxyl. Alright, because notice here, you have this nucleotide attached to this one. Alright, well, how are these two things attached? Well, they're attached through a covalent bond here. We call it a phosphodiester bond. Alright, it's a covalent bond. Alright, so they're covalently linked. Alright, so the phosphate attaches to the 3' carbon of the pre-existing nucleotide. Alright, so talking about complementary base pairing. Alright, so there are these rules called Chargaff's rules. And basically, when you're talking about a molecule of DNA, or you're talking about a molecule of RNA, alright, the amount of adenine in a molecule of DNA is equivalent to the amount of thymine. Alright, the amount of guanine is equivalent to the amount of cytosine in a molecule of DNA. Alright, so what that means is, alright, for instance, if let's say that 25% of the DNA is composed of adenine. Alright, well, you have an equal amount of thymine, so that means that 25% is composed of thymine. Alright, so that means that guanine and cytosine comprise a total of 50%. What about for each? Well, basically, the amount of guanine is equivalent to the amount of cytosine, so you basically divide that by half. So the amount of guanine is 25%. Alright, the amount of cytosine is 25%. Now looking at complementary base pairing, adenine is always pairs with thymine, guanine always pairs with cytosine. Alright, so this has to do with the ability of these particular bases to form hydrogen bonds with one another. Alright, so adenine forms hydrogen bonds with thymine. Alright, so they have the ability to form hydrogen bonds, 2 hydrogen bonds. Alright, guanine and cytosine form 3 hydrogen bonds. Alright, it's always a purine with a pyrimidine. Alright, it's never going to be a purine and a purine or a pyrimidine and pyrimidine. Alright, because if it's a purine and a purine, the molecules are too close together to form these hydrogen bonds. They're like right on top of one another. There's not enough space for them to move and shift. And if it's a pyrimidine and a pyrimidine, they're too far apart. Alright, so it's always a purine and a pyrimidine. Alright, because they have to be able to form these hydrogen bonds. Alright, so adenine and thymine form double bonds, guanine and cytosine form triple bond in terms of hydrogens. Alright, so looking at a molecule of DNA. Alright, so DNA, alright, is double-stranded. Alright, so you have 2 strands of polynucleotides. And these polynucleotides are oriented in opposite directions. Alright, so the fact that they're oriented in opposite directions, because you have one going in this direction and the other going the opposite direction. They are anti-parallel. Alright, so you have complementary base pairing between the 2 polynucleotides, 2 strands. Adenine pairs with thymine, guanine pairs with cytosine. And then the molecule of DNA, you get this right-handed twist, and so you get this helical structure to the DNA.
DNA consists of 2 complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the two sugar phosphate.
Alright, so here you're being shown, again, that short little animation kind of gave you an overview of the structure of DNA. Alright, so it showed you a space filling model. Alright, so here this is showing you the two strands of DNA. You have the one strand going in this direction, the other strand going in the opposite direction. Alright, notice here at this end you have a free 5' phosphate, at this end you have a free 3' hydroxyl. But on the other end, you have a free 5' phosphate, and the opposite end, free 3' hydroxyl. Alright, and then you have complementary base pairing between your bases. Alright, so looking at the structure of chromosomes in terms of comparing prokaryotic and eukaryotic chromosome structure. Alright, so eukaryotic chromosomes, alright, they are going to be linear. Alright, they're helical in nature, and you usually have more than one chromosome. Alright, so your DNA wraps around proteins called histone proteins. And so the DNA, as it coils around these histone proteins, it makes up something called a nucleosome, all right, which is basically the DNA and a histone protein. And these nucleosomes, they'll collectively coalesce together, and they make up what we call the chromatin. This chromatin can condense and become more coiled and will form the chromosome when it's condensed. Now, when you're talking about prokaryotic chromosomes, all right, the prokaryotic genetic material, recall that it's not contained within a nucleus. All right, so it's actually going to be attached to the cell membrane of the bacterial cell. All right, now there are DNA binding proteins. All right, there are histone-like quote-unquote proteins that the DNA wraps around. All right, so it can be in this condensed or relaxed state. Now, when you're talking about a bacterial chromosome, bacterial chromosomes are not linear. At least most of them are not. They are circular chromosomes. And you usually only have one nuclear chromosome in a bacterial cell. Now, this bacterial chromosome can either be relaxed, all right, or it can be supercoiled. And so it can become even more tightly supercoiled when it interacts with these DNA binding proteins. Now, comparing coiled, supercoiled, and relaxed DNA chromosome in terms of the bacterial chromosome. Alright, so a relaxed open circle chromosome. Alright, you have this complete oval shape. Now, when you start to kind of twist the DNA molecule, well, how do you do that? What happens is the chromosome you have to have a break in the chromosome, a double strand break, and it has to reattach around the opposite side of the DNA, so it forms this loop structure and becomes twisted. Alright, so it's kind of like a telephone cord, an old telephone cord. Alright, it has to basically intertwine and wrap around and twist up in on itself. Alright, the more twisted it becomes, the more tightly coiled it becomes, the more supercoiled. Alright, so here you have a relaxed chromosome. Actually, here, it's not really a chromosome. In this case, you're looking at something very similar to a nuclear chromosome, but in this case, you're looking at a plasmid. Alright, because it would be really way too big to look at the entire bacterial chromosome and look at a relaxed versus a supercoiled portion of it. Alright, so here you're looking at a plasmid. Alright, so this is a relaxed plasmid, and this is a supercoiled plasmid. Alright, so what about the genome size? Alright, so compare eukaryotic genomes to prokaryotic. Alright, well, you can already tell their eukaryotic chromosomes are much, much larger in terms of size. Alright, bacterial chromosomes, prokaryotic chromosomes are much smaller. Alright, and then there's varying degrees within each in terms of the size of the genome. Now, looking at a bacterial chromosome, nuclear chromosome, versus a plasmid. Alright, so recall that bacterial cells have extraneous chromosomal DNA that we call plasmids. Alright, now looking at the nuclear DNA, alright, it encodes for various genes. Alright, so here, alright, the ORI seed site, alright, this is the origin of replication. Alright, so when we start talking about DNA replication, the ORI C is where DNA replication begins. Alright, now you have all these other types of genes. You have genes that encode for the ability to break down certain types of nutrients. You have genes that are involved in sporulation. You have genes that are involved in ability to utilize a certain resource from the environment. You have genes that are involved in DNA replication and transcription and translation. And notice how the genes are organized. Alright, you have what's referred to as an operon. Alright, so genes aren't spread out randomly throughout the bacterial chromosome. What you see is that genes are organized because they are related to one another. Alright, so like here you have a lac operon. Well, the genes that are involved for lactose degradation, you have those four genes are grouped together. Alright, so I, Z, Y, and A here. Alright, so for high frequency recombination, alright, you have these genes that are organized together. For, let's see here, tryptophan biosynthesis, we have these genes that are organized together. For histidine biosynthesis, these genes are organized together. Alright, so genes are organized into operons in bacterial cells. Alright, so they're much easier to coordinate and regulate the activity of those sets of genes. Alright, they're not spread out randomly throughout the chromosome. And since, they're not eukaryotic chromosomes, all the genes are found on one single chromosome, not many, like you would have on a eukaryotic chromosome. Alright. Now, plasmids encode for kind of extraneous beneficial factors that's bacterial cell. So things like resistance to heavy metal toxicity. All right, you have, for instance, catalase production. You have genes that are involved in the replication of the plasmid itself. And your plasmid has its own origin of replication called the Ori T site, or the origin of replication for the plasmid. Alright, plasmids can encode for factors that are involved in antibiotic resistance, or the ability to take up and utilize a specific nutrient resource from the environment. Alright, so look at the size of the bacterial chromosome compared to the plasmid. Alright, so this here is showing you E. coli. Alright, so all these little squiggly lines, this is showing you the bacterial chromosome. Now, earlier I was talking about supercoiled and open circular or relaxed DNA. You'll notice this segment of the chromosome is highly supercoiled. This segment is kind of relaxed. It's open. Alright. So think about it. If it's supercoiled, more than likely that part of the genome is not being transcribed or translated. Alright. If it's open, that means those genes are open to transcription. Alright, and other factors for regulation. Now look at these smaller pieces of genomic material. Here, 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 here. Alright? All these are plasmids. Alright, so they're much, much smaller in size.